Hello. Today I would like to talk about a few things we've been doing in my research group. The title of my talk is Efficient Algorithm and Computational Method for Biological Data Analysis. So my name is Shaojie Zhang. So I'm a professor at the Department of Computer Science. So my primary research is computation biology and bioinformatics. Another is an algorithm design and implementation. The primary teaching is Introduction to Discrete Structures, Design and Analysis of Algorithm, Introduction to Bioinformatics, and Algorithm in Computing Biology and Computation Genomics. Currently, my group are funded by the NIH and NSF. So I have graduated 7 PhD students and 2 master thesis students. So currently there are 9 PhD students in my group. First, I would like to give an overview of my research, then I will provide 3 research highlights. So the theme of my research is efficient algorithm design for biological data analysis. First project we are working on is a computer method for non-coding RNA analysis and discovery. So we want to identify RNA structure motif in RNA structures. And we also work on RNA structure alignment and non-coding RNA gene discovery. A third one is NGS-based RNA structure probing and RNA interaction and RNA protein interactions. Last one is RNA design, which is design RNA based on the knowledge we learned from previous topics. And of course, make sure the design is stable enough. We also interested in studying the RNA folding landscape. The second topic in our group is efficient algorithm for genealogical search and the biobanks data analysis. So basically, we want to design algorithm and method for fast detection of IBD segment. And we already using our tool to analyze the IBD segment in the UCF biobank data. So we also proposed a lot of efficient algorithms for genealogical search. The third topic in my group is also design efficient algorithm, but now it's not for biological data, it's for the gate level netlist analysis. So we've been working on this for the last few years. I have a tool set called NETA, which include the ReFSM, ReLIC, RePass, ReRTL, and ReLIC-Find. So here is the one diagram showing the ASP.NET 2019 to show how to use NETA to for netlist analysis and the functionality reconstruction. So since the title of my talk is computer method for biological data analysis, I will pick up 3 highlights out of first two topics. So highlight number 1 is RNA structure motif identification. So this project has been funded by NIH since 2012. So we have developed a series of bioinformatics tools for RNA structure motif identification, discovery, and classification. We also use our tool to discover many novel structure motifs. So here show an example of structure motif, which is Kinter motif. You can see there is a lot of non-canonical interaction in the loop regions. So basically, we have been developing many algorithms and tools try to analyze such structure motif. Here's an example of one of the tools called RMotif scan, which can convert the motif identification problem into click finding problem. So here's another example show a new motif we discovered using our tools. So here show 2 multi-loop region. One is in the ribosome RNA, another in twister enzyme. The future for this project is we try to build a database for all the known motif, new motif and existing motif we've been discovered, And so that we provide that to the biologists or biomedical scientists. They can use the motif to design new RNA, also try to understand their functionalities. So highlight number 2 is sgRNA design for CRISPR-based labeling. So this week, the Nobel Prize in Chemistry was awarded to the technology for CRISPR-based gene editing. But for the product we are working on is using them to labeling the DNA so that we can see the movement of DNA in the cell. So this platform was developed by our collaborator at the UMass Medical School. So my lab contribution, there are 2 major contributions. First one, we designed the sgRNA guide sequence such that they can target the chromosomal specific repeat in the human genome, so that makes the signal stronger. So that's our first paper published in 2015. Another example is to make the signal even stronger, we designed the sgRNA scaffold, make sure they're so that there are more RNA aptamer to bind the fluorescent proteins. So make the signal even stronger. And here's the pipeline how to design such an RNA. So you can see we have to think a lot when we design such RNA so that they can follow the correct second structure. Also make sure all these aptamer are not interfering to each other. So this platform can be useful to investigate the DNA movement in itself and the chromosome dynamics. So third highlight is the efficient algorithm for genealogical search and biobank scale genetics. So this project is also funded by an NIH grant. So what's IBD? IBD stands for identical by descent. So basically, are the identical chromosomal segment shared between 2 individuals due to their common ancestors. So we have developed a few computing tools for fast detection of IBD segment in the biobank scale cohorts. So this figure shows one of the tools called Rapid, which published last year in Genome Biology. So to use the Rapid, we call all the IBDs and analyze them in the UK Biobank data. So UK Biobank is a consortium with 500,000 volunteers from UK with their genetic and health data. So our preliminary result already uploaded to bioRxiv. So here's some highlight. So we found 3.5 billion IBD larger than 5 centimorgan among the UK biobank participants. So 5 centimorgan equivalent to around 5 million bases. And so interesting, for each participant, they have around 14,000 relatives, which means they share at least 1 IBD. To extrapolate that, for each UK person, they will have about 3% of the UK population connect to them. And here the figure to show you is an interesting finding is basically the people live in central London, which is near the Manchester region, has more relative than people live in southern England. Sorry, central England to southern England, which is London region. So one thing is for us to let you know is Rapid can generate 3.5 billion IBD result in less than a CPU week. So we can see that IBD can also be used for genealogical search. So many DTC companies offer the genealogical search to their customers. Right now, there are more than 30 million customers already, get their data for the family for the relative matching. So for this particular direction, we have developed several efficient long match query algorithm so that that can enable IBD-based fast online genealogy search. Recently, we published a paper in Recom. So we developed the first dynamic PPW data structure, allow efficient updating the database. So in this case, our new algorithm can make the genealogical search easily to scale up for millions of individuals. So for the future work, we can see that there are many people got their DNA sequenced. So with our efficient informatics method for IBD detection, I hope we can build population-based genetic relationship. With that, there are a lot of new possibilities for population genetics, genealogy search, and disease genetics. So for collaboration, we are interested in how to, employ our new tool to the cloud, and we also need some help regarding the, security and the privacy issue when we dealing with the genetic data. Okay, so finally, I would like to thank my students who has been working on this project and our collaborators, Dr. De Guizhi and Dr. Xiaoming Li. We are working on the IBD-based identification project. Dr. Ma and Dr. Patterson from UMass Medical School work on the CRISPR-based labeling, and Dr. Ye Jin from University of Florida work on the netlist analysis, and finally, Dr. Wenqian Hu from Mayo Clinics. We have project on iron protein interaction. And here's our funding support. And of course, we would like to thank our data results from UK Biobank. Thank you.